Hey folks, we're going to do a video today about the male reproductive gross anatomy. So what we have here is a section of the male pelvis where we can see both internal and external things. Let's start with the external stuff. In this male pelvis, we can see a lot of things including the scrotum. The scrotum is going to house basically two pairs of major organs. Of course, there are the testes. There is a testy right there, that whitish thing. That testy is the site of production of two important things, one cellular, one hormonal. The cellular product of the testy are sperm cells, and the hormonal product of the testy is going to be the testosterone, the male sex hormone. Both of those things are produced pretty much from puberty until death do us part. Now, sitting kind of superior to the testy, we see an epididymis. I'm going to spell that one just because it is a little bit more challenging of a word, if I can spell it correctly. Epididymis. Now, just for fun, realize the plural for epididymis is epididymides. Epididymides. It's kind of fun to say. Now, here's the deal about sperm cells. When sperm cells are made in the testy, they do not yet have the ability to swim. They are pushed by the pressure of testicular fluid into the epididymis, and the epididymis is the site of sperm cell maturation and sperm cell storage. If sperm are not ejaculated from the epididymis, then they will simply be reabsorbed, chopped up, and recycled so that their pieces can be used again. All right. Now notice that we have the testy and the scrotum, which is a rather unsecure, unprotected place. Why do we do this? Why do we put these valuable organs, the testes, in a place that is protected by neither bone nor muscle? Well, the reason is pretty simple. Actually, it's not. It's kind of complex. There are several reasons. One of them is temperature. So the temperature is lower in the scrotum than it would be in the internal body cavity, and that is going to enhance sperm production. That's a human reason right there. Now, in other animal species, the presence of a scrotum is often used as a means of male display and mate attraction. Hopefully that does not hold true for human beings. Okay, so what else do we have here? Coming up from the epididymis, we see a tube. That tube is the Vas Deferens. There are two Vas Deferences. The Vas Deferens will carry, by means of peristalsis, sperm cells plus testicular fluid, epididymal fluid, onward towards the urethra during an ejaculatory event. Now, the Vas Deferens has some neighbors. We see the testicular artery. We see the testicular vein. And there are also nerves and lymphatic vessels that would be within that mix. And the combination of all those things is referred to as the spermatic cord. So we see a little chunk of the spermatic cord. Now, all the structures of the spermatic cord are going to come up and pierce the abdominal wall. They're going to pierce the abdominal wall right here. There is a weak spot in the abdominal wall on either side, just superior to the pubic bone, and those weak spots are known as the inguinal. Remember, that's the groin region, the inguinal canals. The Vas deferens then continues over the urinary bladder, which we see right here. The Vas deferens goes past a ureter, and we see a ureter right there. And don't forget, there is another ureter hanging out over there. The Vas deferens will then go next to the seminal vesicle. And when we chop this model up, we'll see what happens next. Now, there are two seminal vesicles. We see one right here. The two seminal vesicles make seminal fluid, which is a basic fluid. It comprises 60% of semen volume. It is rich in chemicals necessary for sperm activation and also rich in nutrients for the sperm cells to feed themselves upon as they progress through the female reproductive tract in hopes of finding a secondary oocyte to fertilize. Now, what else can we see here? We can see the prostate gland, which makes another 30% of sperm volume. Not of sperm volume, I'm sorry, of semen volume. Again, a basic solution, rich in nutrients and necessary chemicals. Now, if you notice, the seminal vesicles make 60% of semen volume. The prostate gland makes 30%. 30 plus 60 is 90. The other 10 comes from the seminiferous tubules by way of the epididymis. So we're talking testicular fluid and sperm cells. All right, let's clear this all up and point out a few more things. We can also see the urogenital diaphragm, which is part of a broader pelvic diaphragm. So there would be a whole layer of muscle within the pelvis here. We see the part immediately beneath the prostate gland, which is the urogenital diaphragm. We also see the penis. There is, of course, the shaft of the penis. There is the glans penis, and this penis has been sliced. So we see the foreskin, sort of ish, on one side. There it is. The foreskin or the prepuce is visible right there. All right, I think that is it for this particular model. So let's, well, not this particular view. Let's slice it open and see what we have on the interior. Okay, what do we got? What's going on here? We have the coiled tubes of the epididymis within the scrotum. We have the coiled lobules of the seminiferous tubules in the testes. That is where sperm production is going to take place. We also see the interior of the penis. We see some erectile structures. That one that I just put a pink arrow on, that one is one of the two corpora cavernosi, rather. One is simply a corpus cavernosa, and there are two of them. We see one right here. We also see another erectile structure. We see another erectile structure, which is this guy. Oh, where's my arrow? Come on, arrow. Let's get a red arrow there. Which is this guy right here. And we see him on both sides of the urethra. Do notice that that is the penile or spongy urethra that we can see. Let's put an arrow on that penile or spongy urethra as well. And we can see an erectile tissue flanking it, rather encircling it. That would be the better word. That erectile tissue is singular. There is only one of them. It is the corpus spongiosum. So all three of the erectile tissues, two corpora cavernosi, one corpus spongiosum, they are riddled with vascular spaces, which can become engorged with blood. So arterial dilation happens. Blood rushes in. Penile venules are actually compressed by the arterioles as they dilate. It's easy for blood to get in the penis. It's hard for blood to get out. And that results in an erection. Do notice that the corpus spongiosum is going to expand distally, not so much on the ventral side, but more so on the dorsal side, to form the glans penis. All right, there is more that we can see in this particular view here. For example, we can see the prepuce covering the glans penis right there and there. We can see the urogenital diaphragm again, just sliced open. We can see the membranous urethra running through it. And the muscle on either side of that membranous urethra, right here and right here, is the external urethral sphincter. All right, see you in the next one. Bye-bye.